Hello and welcome to the World Advanced Therapies and Regenerative Medicine Congress Interview Zone. My name is Freya Lees, I'm the editor of RegMedNet and I'll be your host for today. Now I'm very pleased to be joined by Jeff Kutok from Infinity Pharmaceuticals. Jeff, thank you for joining me today.
Thank you for having me, it's great to be here.
You're very welcome. So for those of us who might not be familiar with Infinity Pharmaceuticals, can you give us a little bit of background on the company?
Sure. Infinity is actually a company that's was founded in the early 2000s, so it's been around for quite a while, and we have a focus on small molecule inhibitors of kinases. And our most recent program, and the one that we're focusing all of our attention on, is an inhibitor, a small molecule once-a-day oral inhibitor of PI3K gamma in the immune suppressive macrophages and MDSCs that prevent T-cell responses in tumors. So through targeting of that immune suppressive macrophage or myeloid cell, we feel that we can enhance responses to some of the therapies that are becoming more standard today, like the checkpoint inhibitors.
Mm-hmm. And how did you yourself come to work with Infinity?
I came from academia. I was a professor and a clinician in the Harvard system, which was nearby our Cambridge, Massachusetts offices, and I had a long interest in translational medicine and biomarker work, and I was recruited to the company about 8 or 9 years ago to develop their translational science program, and since then I've been involved in a variety of different programs, and the program that we're currently exploring now which is a small molecule inhibitor, IPI-549, is one that we developed internally through our own research, and we've brought it all the way from the preclinical stages all the way to the clinic now, and we're in phase 1 and soon to be in phase 2 studies.
Mm-hmm. Fantastic. So obviously checkpoint inhibitors now are a fairly well-established therapeutic,
Sure.
but they weren't always. So can you tell me how you think the immunotherapy industry has developed since the discovery of checkpoint inhibitors was made?
Sure. There's no question that the checkpoint inhibitors have been transformative in oncology therapies, and there's hundreds of thousands of patients who've benefited from the checkpoint inhibitors. But I think what's sometimes less well appreciated is that not everyone responds to a checkpoint inhibitor. The response rates to the monotherapies are in the 20 to 30% range. And some patients, there are some tumor types that just don't show any responses whatsoever. So our company was focused on developing ways to overcome that resistance, inherent resistance to the checkpoint inhibitors. And we uncovered a role in our work exploring the PI3K family of kinases for PI3K gamma, which has this very unique and specific role in the immune suppressive macrophages and myeloid cells that infiltrate a tumor and act to suppress immune responses. And they can do that even in the setting where a checkpoint inhibitor is present. These cells produce a variety of different cytokines that are important in suppressing responses. And we uncovered in a series of experiments were published in the journal Nature in 2016 with our academic collaborators that PI3K gamma signaling is a key mediator of this immune suppressive phenotype in these cells, and that if you block that, you not only remove that immune suppression, but you actually reprogram the cell to become a pro-inflammatory cell type, and it contributes to the ongoing immune responses. So we're focused on that program, and we were also were able to determine preclinically that in tumor types that were completely resistant to a checkpoint inhibitor, when you add our compound and overcome that tumor microenvironment resistance, you can now reactivate a response to the checkpoint inhibitor, and we're exploring that clinical setting in our phase 1 studies.
Exciting. So when developers have created their wonderful therapy, it's obviously tempting to try and maximize value by expanding indications. We've already seen it with Kymriah and Yescarta. Why is it important to try and expand these indications, and what are some of the challenges in doing so?
I think the real challenge is to understand the science and the biology behind why certain indications, and even within an indication, certain patients' own tumors are not responding to either checkpoint inhibitors or other forms of immune therapies. And I think that in order to really go beyond where we're at currently, we have to really use all the tools possible to understand within a given tumor's environment, what are the drivers behind the suppression of an immune response. I think we can all agree that that immune surveillance during the early parts of a tumor's life is critical in keeping us relatively cancer-free. But once tumors begin to get established, there are a host of mechanisms that take place in the setting of any immune response to dampen and suppress that immune response. We have to understand in patients given tumors, what are the drivers behind that immune suppression. And then when we understand that, we can go to our tool chest and our medicine cabinet and decide which therapies are really necessary to be combined and given to a patient to specifically overcome that immune response and immune suppression in that patient and affect a better anti-tumor immune response.
Interesting, thank you. So with these immunotherapies, a lot of advanced therapies, regenerative medicines, the patient populations are really small in these rare diseases. So it's really important to really prove that your developing therapy is effective. What technologies can developers try to use to really show how great their therapy is?
So you mean beyond just a clinical response?
Yeah.
I think it's an important point that you really do need good biomarkers of biological activity. It's particularly true with many of these modifiers of an immune response, including our agent, which modifies the suppressive response of a myeloid cell, because they may not in and of themselves be active. They may require combination therapies. So in order to really determine whether your drug is doing what you think it is, you really do need biological markers of immune modulation in these tumors. So and that comes in the form of looking at changes in the immune phenotype of cells, using flow cytometry, looking at the changes in gene expression, and then or looking at generalized markers of immune activation, including things like gamma interferon responses and upregulation of certain cytokines that are important in inflammatory responses. So I think that will help really get a better sense that your drug is doing what you're hoping it will do. And even in the setting where you're looking in phase 1 patients that are less responsive to any therapies in general.
Fantastic. So circling back to Infinity Pharmaceuticals, you mentioned a little earlier on that you have some potential advancements in sort of preclinical testing, clinical testing. What can we look forward to from Infinity in the next few years?
So right now we're in the later stages of our phase 1 study, which was designed to evaluate the effect of our drug IPI-549 as a monotherapy in advanced solid tumors, and in addition to evaluate how well it combined with an immune checkpoint inhibitor. We're partnering with Bristol Myers Squibb and their anti-PD-1 antibody nivolumab to look at that. And we're already through our dose escalation, and we're into our expansion phases for the combination studies. And there we focused on a patient population that had a very high bar for responses. We're looking in patients who have become resistant to a checkpoint inhibitor as their immediate prior therapy.
Yeah.
And then we're continuing the checkpoint inhibitor, nivolumab in this case, and we're adding our antibody, our IPI-549, our small molecule inhibitor, to see if we can augment an immune response in these patients that are already resistant to the checkpoint. And we have some promising translational and clinical data with respect to the phase 1 that we presented publicly. And the idea there is that we feel that we have strong enough proof of concept to move on to later phase studies, including a phase 2 study that's a randomized global study comparing response of nivolumab plus IPI-549 to just nivolumab alone in bladder cancer patients who become resistant to platinum therapies. And we're excited about that. And there we're focusing on a group of patients that have high levels of circulating myeloid-derived suppressor cells. This is the target cell of IPI-549, and we're enriching for patients with high levels in the hopes that we can overcome that immune suppression and have a better response with respect to the combination. We're also moving up into the front line with a collaboration with Roche Genentech, where we're looking in triple combinations, including Abraxane plus atezolizumab, their anti-PD-L1 antibody, plus 549 in triple-negative breast cancer in the frontline patients. And we're combining IPI-549 with Avastin and atezolizumab in the frontline in renal cell cancer patients. So we're moving sort of in greater breadth with more combinations, more unique combinations, and greater depth with respect to the line of therapy, moving from these very resistant patients to patients in the earlier lines of therapy. And we're also, I mentioned one other thing, we're also looking with a triple combination in collaboration with a company called Arcus Biosciences, who has a adenosine receptor antagonist. And there we're looking at a checkpoint inhibitor-free regimen. We're combining IPI-549 in combination with their adenosine receptor antagonist and a chemotherapy in triple-negative breast cancer. So that's what we have to look forward to. These later phase studies are beginning to kick off, and we're hopeful that by the end of 2019, all of these studies will be underway.
Well, I've got everything crossed for you, Jeff. Thank you so much for joining me today.
Thanks so much for having me. This was great.
Thank you, and thank you all for joining us. We'll be back with more interviews from the Interview Zone very shortly.